Before we take our first breath of air, our DNA and genes are already determined. From our hair color to the way we act in different settings, the collection of all our genes, also known as the genomic structure, creates the blueprint for all we are and will be. Although our genes can be affected by our environment as we grow, which is described as epigenetics, our physical traits are less flexible. Even though cosmetic surgeries do exist to change the physical structure of our bodies, what if we could change the structure at its core, its genetic structure? It's Agent RX, and welcome back to episode 3 of The Next Prescription. Today, we will be exploring the rapidly developing technology of Crispr-Cas9, an upcoming major player in the field of gene editing. Today, we will explore what a restriction enzyme is, what Crispr-Cas9 is, its evolution, the future outlooks of this new technology, and the ethical concerns associated with it. What is a restriction enzyme? Before we can understand the process of Crispr-Cas9, we first must explore what a restriction enzyme is. An enzyme is a type of protein. A restriction enzyme is a specific protein that cuts DNA. The restriction enzyme will locate a specific sequence of nucleotide bases, adenine, cytosine, thymine, and guanine, and make an incision there. The restriction enzyme can make two kinds of cuts, a sticky and blunt cut. The sticky cut is where the restriction enzyme makes a staggered cut of the DNA. One of the strands is longer than the other. Some enzymes that make a cut like this are EcoRI and BAM-HI. On the contrary, blunt cuts cut the DNA at the same vertical longitude, making the ends of the DNA strands straight. Some enzymes that perform blunt cuts are small and EcoRV. Although both cuts are important in the scientific world, sticky cuts are more important when it comes to gene editing. The natural use of restriction enzymes only happens in bacteria, which is also where all restriction enzymes scientists use are found. Scientists are able to extract the enzyme and use them in the laboratory to alter DNA fragments. The restriction enzymes will cut at a specific palindromic site. A palindromic site is a site where the length of DNA reads the same backwards and forwards. An example would be GAATTC, or in the English language, race car. Both sticky and blunt ends are used, but sticky ends are more efficient as blunt ends require the help of another enzyme known as DNA ligase, which acts as a glue for the two DNA sequences. Restriction enzymes are the basis of CRISPR-Cas9. It's just a more advanced application of it. From tiny proteins inside bacteria to the ability to change certain DNA sequences in our bodies, it is a fascinating story as to how scientists were able to create CRISPR-Cas9. Before its history encapsulates us, we first need to understand what CRISPR-Cas9 really is and how it works. What is CRISPR-Cas9? CRISPR-Cas9 is a relatively new gene editing tool that could change our genetic structure. In theory, with enough time and tools, the height of a person or the color of one's eyes could be changed. Although those ideas are extremely far-fetched and unlikely, CRISPR-Cas9 has still been making small changes to our genomic structure. CRISPR, or clustered regularly interspaced palindromic repeats, make it possible to quickly change the expression of genes by turning them on or off and correcting mutations formed during DNA replication. CRISPR involves three main components: the Cas9 protein, a guide RNA, and the targeted DNA. To start off, let me make it clear. Cas9 is not a restriction enzyme, but it operates similarly given that both restriction enzymes and Cas9 are endonucleases. Endonucleases are a broader category that restriction enzymes fall under. They are enzymes that cleave DNA at a specific site, not necessarily at a palindrome like restriction enzymes require. Unlike restriction enzymes, Cas9 is programmable. Cas9 enzymes cut at a site encoded into it by a guide RNA, a single-stranded piece of DNA. Cas9 is like a GPS. I'm sure you know how to operate a GPS. You input a destination and it guides you to it. That is similar to how Cas9 works. The GPS is the Cas9 endonuclease, doing all the operating. The guide RNA is the inputted location, telling you where to go. The destination is the DNA site that needs to be cleaved. In a lab, scientists will attach the guide RNA to Cas9 and release it into the DNA strand. From there, Cas9 will locate and cleave the corresponding 18 to 20 nucleotide sequence presented on the DNA. After that, Cas9 has one of two options: knock in or knock out the gene. Knocking out a gene requires the deleting or inserting of nucleotides, which is a non-homologous repair pathway, or NHEG. Knocking in a gene is more difficult as it requires a substitution of a new DNA sequence known as homology-directed repair, or HDR. The HDR method is more accurate, however, it is less efficient due to the narrow window of time there is to perform the operation. CRISPR is a simple concept to understand, but the story as to how we reached this remarkable technology is long, exciting, and screaming to be told. Let's dive in. Beginnings to modern CRISPR-Cas9. CRISPR-Cas9 is a convoluted and godlike process. It's hard to imagine that it all started with one of the simplest forms of life, bacteria. Bacteria is constantly being bombarded by phages, which are special viruses targeting purely bacteria. Like a virus, the phage inserts its DNA into the bacteria. The bacteria unknowingly nurtures the phage to replicate until the bacteria bursts, releasing many new phages into the environment. In order for bacteria to sustain themselves, defense mechanisms had to be developed, one of those being CRISPR. CRISPR captures sections of the invading phage and inserts them into the genomic structure of the bacteria. This allows the genome of the bacteria to memorize the harmful phage sequences and build immunity to them. Now, the previously unsuspectable phage can be detected and destroyed thanks to CRISPR. And that brings us to the name, CRISPR, clustered regularly interspaced palindromic repeats. The acronym was chosen because of the direct observation made by scientists when observing bacterial DNA. They found clusters of palindromic repeats, about 30 base pairs long. These clusters of DNA were often separated by another section of DNA known as spacers. This is where the DNA alterations for immunity of phages were performed. The first hint of this bacterial activity was discovered in 1987 by Japanese scientist Yoshishima Ishino. While observing E. coli, he saw unusual repeated sequences of DNA pieces embedded into a larger fragment of DNA. Although CRISPR was technically discovered by Dr. Ishino first, the discovery is credited to a Spanish scientist known as Francisco Mojica. He was able to piece together how CRISPR worked and gave it its name. He was observing archaea, which has a similar problem and response to viruses as bacteria, when he too noticed the same repeated sequences as Ishino. Mojica was able to figure out the sequence matched the genome of bacterial phages. This helped him conclude that there was an adaptive bacterial immune system in place, which he called CRISPR. After understanding how CRISPR worked in bacteria, it was hypothesized by Dr. Jennifer Doudna and Dr. Emmanuel Charpentier that CRISPR could be repurposed as a gene editing tool. As they researched together, they figured out their hypothesis was in fact valid and published their findings in 2012 to the journal Science. Only a whole 8 years later did Dr. Doudna and Dr. Charpentier receive a Nobel Prize in chemistry for their discovery. Not only a monumental moment in science, but also in the media, as they were the first all-female team to win a Nobel Prize. In 2019, CRISPR-Cas9 started to be used in clinical trials, and as of 2020 is being injected directly into human patients as gene therapy. But there is still so much more for scientists to work towards. what the future holds. CRISPR has slowly released itself into human trials and clinical studies, and with success screaming all around the board, gene editing to cure human diseases is feeling much more like a reality rather than fiction. CRISPR could be used to change the genomic structure of not just our regular genomic structure, but our epigenome, the genes that are affected by our environment, too. It is also providing hope into changing genetic disorders that are assigned at birth. Before, these disorders were thought to be unchangeable, but CRISPR is shedding new hope. In a trial conducted in 2021, CRISPR-Cas9 was injected into the human bloodstream of individuals born with a fatal disease causing their heart and nerves to shut off. CRISPR-Cas9 was able to shut off the toxic proteins being produced by the liver, which has led doctors and scientists all over the world to see the power of this tool. How this tiny tool can permanently cure a life-threatening disease in a one-time procedure. But CRISPR-Cas9 is not just holding a stance in the world of human biology, but the one of plants too. CRISPR is allowing for new ways to genetically modify plants. CRISPR-Cas9 is allowing for plants to be drought and disease resistant as never before. Some speculate that in only a few years, CRISPR-modified plants and food will be available for consumption. Ethical concerns. When a technology that has the power to change the structure we were born with on a micro level enters the world, there is no shock that ethical questions arise. One concern is the ability for the Cas9 protein to cause unintended side effects. As you know, Cas9 uses a guide RNA to locate the target gene. But what if it drifts to another gene and edits there instead? An off-target effect. Even if it arrives at the proper gene, what if it makes a faulty edit there? An on-target effect. Both of these mistakes have the potential to harm the health of the organism, whether it be human or plant. They can create unwanted phenotypes, the expression of a gene like eye color, in the environment and mess up the society of the natural world. Even though these unintended edits could lead to unprecedented advantages or have no effect on the individual, it is important to ask, when do the potential benefits justify the risk of unintended or unwanted outcomes, according to the Innovative Genomic Institute? Another concern is the worsening of inequality between rich and poor. CRISPR is a complex treatment and technology that is primarily funded and researched in wealthy Western countries. This can allow for Western countries to achieve an even greater dominance on the rest of the world. Even within those countries, the procedure is costing around $2 million, so only the wealthiest of those countries can afford it. Right now, CRISPR is currently working only on diseases that have nothing to do with the germ cells, sperm and egg, meaning diseases that aren't passed down. However, CRISPR-Cas9 research is expanding into genetically passed down diseases like familial Alzheimer's, a type of inherited Alzheimer's. This means CRISPR has the ability to remove diseases from a whole bloodline of families, particularly the wealthy ones. CRISPR can exponentially increase the differences in wealth and health already present in today's global society. One of the most ethically challenged is how CRISPR-Cas9 allows us to play God. People with religious views believe that God should be the only one who can change the ways of nature, while others who don't believe in God see CRISPR as nothing but a new way to put good into the world, to bring more efficient and productive members to society. The difference in views are raising questions as who should decide if we can edit genes? Is it our place to do so or a supernatural force? The issue is simple and easy to understand, yet the simplicity is underlined with a matter closely tied to people's hearts, making it a difficult one to overlook. Conclusion. Despite all the ethical debates on CRISPR, scientists are continuing to push through on their research on the topic, expanding to learning about germ cells and finding cure to genetic diseases to learning how to treat life-threatening diseases like cancer. It's one of the newest and most promising biotechnologies emerging in the world, giving hope to change health complications all over the world. CRISPR-Cas9's rapid growth makes it crucial for ordinary individuals like you and me to be informed about its power and potential effects. I'd love to hear your thoughts on this revolutionary technology entering our world in the comments. Thank you for tuning in to the next prescription. And until next time, this is Agent RX signing off.